Okay, so I'm back and let's start our discussion. So, 1 minute, recording started. So now we are actually shifting little bit the topic of discussion. As I told you, like before, in the yesterday night, I also posted one sentence in the MS Teams that we will discuss about lot of things today. So, but just fundamental idea. You don't need to remember all those complex names and other things. I'm trying to avoid those also. How do so many types of cells arise in our body? That is a big question, right? So after fertilization, we have the zygote. When cell divides, then you can see kind of a mass of cells here, many cells together. That is called, some modular stage or whatever it is, some kind of mass of cells together, like around say 60 cells, for example, like approximately something like that. So, but those cells, then what happens? Those cells need to be differentiated. Differentiated means those cells should now acquire little bit of different properties so that they can give rise to different types of cell in our body. And how does that happen? So here in the left side, I am mentioning here that expression of tissue specific genes dictate differentiation path toward a specific cell identity. So as a result of that, if you see here, just very crude and very basic example I am providing here, that during differentiation what happened, say for example these are muscle cells. So as a result of that, what will happen in muscle cells, like genes responsible to make myoglobin, neurotransmitter and hemoglobin, those are three different genes. For example, like hemoglobins are protein, they are coming from some gene. Neurotransmitter can be protein, can be something else also, so they are also coming from some genes. But if you consider in terms of product, the muscle cell, they have myoglobin, but they don't have hemoglobin or neurotransmitter. Now in nerve cells, you don't have myoglobin or hemoglobin because they don't carry oxygen, right? So rather they should have neurotransmitter. So they will transfer signal through the neuron so that we can understand what is happening in the surrounding. So as a result of that, they should have neurotransmitter. On the contrary, in RBC or red blood cell, they should have hemoglobin so that they carry oxygen and that can be reached in different parts of our body. So now the question is how this differentiation happen, how these different function cells acquire. That I would like to discuss in the next slide in a brief manner. Now here on the right side, I'm mentioning that in contrast to tissue specific genes, we have some other genes which are called housekeeping genes. So those are not really terminology. You can mention that housekeeping, you can just remember housekeeping genes means those genes which are required in every cells all the time. Say for example, genes responsible to make tRNA or genes responsible to make RNA polymerase. Those should be the housekeeping genes, because in every cell you need to synthesize protein, you need to make RNA, right? So as a result of that, those are housekeeping genes, right? And those are tissue specific genes that are responsible to make myoglobin, hemoglobin or neurotransmitter. Now, how it happens? I actually mentioned a little bit of that during the lac operon, but here you will get little bit of the kind of idea here, that same genome present in different cells, but they work in a different way. But the differential gene expression in different cells make them different. if you see this example, that this is our DNA, you have 3 genes, just just for explanation, 3 genes here, gene 1 in red, gene 2 here, and gene 3. And those green color stuff, those are a regulatory module. Just like, in case of E. coli, I told you that operator, right? Promoter means where RNA polymerase binds. In addition to that, they have operator. Like that, in our system also, we have more regulatory module. We have more complexities, right? So now, based on the developmental time, space, what happens? So for example, in this, like the gene present in some cells, for example, this is muscle cell. Here you will see that if I tell that this is gene A, B, and C, you are getting product from B and C, but not getting product from A. Although this DNA is same, but in other cell type, you are not getting product from C, but you are getting product from A and B. So that makes these two cells different. This is skin cells and this is muscle cells. We are just showing example of 3 genes, but this is not 3. There are many genes and their permutation combination in terms of expression that makes different types of cells in our body, and that finally make into different system organ in our body required for a complex organism like us, right? And as a result of that, this kind of differentiation happen when we grow up, like from zygote to embryo. When we develop, during that time this takes place. So is it clear?
Excuse me, sir.
Hmm.
Sir, here in the first diagram, only A should get expressed now because transcription regulation are present in the B and C.
Oh, okay, okay. I got it. Yeah. So this is, I understand your point. Uh, yeah. So you are saying that here transcription regulator bind here, so only A will be produced, right?
Yes, sir.
Yeah.
Means,
So yeah, I understand because you are actually comparing it with lac operon. I also told. So lac operon is negatively regulated operon. So where it binds, that then where lactic pressure bind, that protein will not be expressed. But many other genes, it's kind of opposite. So here, what we are trying to say, like I understand your question that this binding, it might activate the gene to make product or it can silence. But in this case, we are showing that wherever the transcriptional regulators bind, then the gene getting activated and it make product. So, but your question is pertinent, no problem. So, in case of lac operon, we explained that when the lactic pressure bind, it inhibits its expression, and you correctly correlate. That's why you asked this question. But in this case, what we are trying to say that transcription regulator, when it binds to the upstream of the gene, then it activates and it make the product. So now if you ask me which one is true, I will say both things are there. Like, so that means these are regulator, right? It might activate gene expression or it may downregulate gene expression. But in this case, we actually demonstrated that if it binds, then it will make product. Okay.
yes, sir. I got for B and C, but for A there is no regulation, so it should always get expressed now.
No, yeah, Rajesh, that's what I'm saying. That, like, this is not your fault, I would say. The fault is like we are showing little bit different image. So again, you are comparing that with the lac operon. That if something is not bound there, that means always transcription will be happening. Rather, I would say in eukaryotic system, it is more complex process. In order to bring RNA polymerase and start transcription, you need various regulator, transcription regulator. First, they will bind so that RNA polymerase come and start the transcription. Then how it feels, Rajesh? Now it's clear.
Yes, sir.
Yeah, so that's why this is very complex process. Here in this case, what I try to say here that in this case, the transcription regulator, you can see those things in a positive way. If transcription regulators are there, then you are getting your genes are expressed. So as a result of that, they will make product. But again, I am telling you, whoever asked this question, this is a good question because you actually recapitulate whatever I discussed in the lab operon. That was a specific case in a prokaryotic system. But here we are trying to say that gene expressions are differentially regulated in different cell types, and that makes those cells are different. And that is how it functions in our body also. Okay, and but not these 3 proteins or 3 genes. There are many genes. They are, as I told you before, more than, like we have around 20,000 protein coding genes, and approximately if you say like 50% of them are housekeeping genes, and the rest of the 50% they are like in some set of cells, some of them off, some of them on. That way we get a huge types of combination. Okay. Now this differentiation.
One
Yeah.
thing. So yes, sir. So the differentiation occurs due to the different transcription regulators occurring in different types of cells. So, sir, in the initial stage, like in the zygote stage, all the cells divide equally. Then where does this differentiation start taking place? Like
Excellent
how,
question. Excellent question. Yeah,
what
very good.
point?
Yeah, very good question. Yeah. So, like we have like 3-4 different reasons actually. Like there are many, but 3-4 actually at this level we can explain, we can discuss, right? Because of time issue, I discarded many of them, but I kept one of them important thing. So that is there in the next slide. So whatever you ask, that is nice question, and logically it should be fitted here. So answer is here. So what you ask, this is the answer of that. I will explain that. 2 ways of making sister cells different. As you asked, that if you consider this is zygote, then after division we will get the exactly same cell. Then how we will see this? The initial difference should be there somewhere. That is Tanmoy's question. I think many of you have
Yes,
the
sir.
same question. Some students are asking. That's okay, but this is the question: that how the initial difference happen, and from there you will see different lineages of cells. So as I told you, it's a very good question. So answer is here. So that depends on like various factors. Here we are mentioning like the first one is asymmetric division. So for example, as I told you, from zygote to you have many cells like 16 cells. For example, 16 cells are there. They are exactly same. Now what happened during division of this cell? Something happened what I'm discussing here. So during the division, the genetic component, they get distributed properly in the 2 cells. Now, apart from the genetic material, there are many proteins, RNA, they are also present in the cytoplasm. Okay. So that molecules like proteins and cytoplasm, they have some preferential distribution. Now, if you ask that why that happened, this will be little difficult for me to explain at this level, but that is happening all the time during the developmental processes. When we get developed, like in one of the image I have shown you that in the first we like zygote to few cell stage, then embryo developing at 4 weeks, 8 weeks. So during that time, this kind of things happening, the preferential distribution. And this is not true only for human. This is true for even a fly, for Drosophila, for fruit fly, for everything. This is happening. For everywhere it is happening. So the one classical explanation is what happened. Some of the proteins which are actually they have some like like some specific properties in terms of their in terms of in context to development, they are like polarized in one part of the cell. As a result of that, when cell is getting divided, this cell here shown in the bottom, they acquire this molecule. It can be protein, it can be RNA. Most of the time it is protein. And this protein has some effect on its DNA. In the next generation onward, it will be diverted into a different direction and it will go into a different direction. So that is one thing is happening everywhere in the multicellular life form, whether it is plant, animal, whether it is fly, human, everywhere it's happening. This is called asymmetric cell division. Again, I am telling you, to all of you, particularly Tanmay, you asked this question. This asymmetric divisions primarily happening during the development process. We are growing inside our mother womb during that time, mostly. Okay. In adult body, in cell, like some scar happen, some cut happen, that time when it gets like cells are getting divided, replenished, that time those are not asymmetric division, those are mostly symmetric division. That means from one cell to two cells you are getting. But this asymmetric division is predominant during the developmental process, and that's why from zygote you are getting a lot of different product, right? Finally by 20 weeks of age, like not age, before birth I'm saying, after fertilization to 20 weeks, by that time, like that baby already have all those different organ and a different system already ready. Okay. So as a result of this is one part, the asymmetric division, and the other one is even after symmetric division, some extrinsic signal, that means the neighboring cells that secreted some signaling molecule. This is little bit more philosophical, I would say, more a little bit complex to explain you. So that also makes some differences. It is kind of extrinsic signal. That means maybe there are many cells together. One of the cell actually helping others to convert, to make it little different. But now you will ask me that cell, why this cell is helping, like induces or providing some signal to this cell. Yeah, so this is very complex, I would say, but this also happens. So those two are the primary reason, either asymmetric cell division, which is very predominant during the developmental stage, as well as some extrinsic signal that helps to make different cell types. And whenever the different cell types form, then you can understand there the expression of genome, expression of genes are also getting separated. As a result of that, finally, we get completely specialized organ. Say, sometime I always say that eye cells, cells present in our eye, those are completely different from cells present in our liver cells or cells in nerves, like, in our brain. So as a result of that, we completely get different set of, although we have same set of information, but different types of function. Okay.
So in the like symmetric cell division, we get the extrinsic signal to convert one of the daughter cells into a different identity. So both, since in symmetric division both are identical in nature, so how do these signals differentiate which
Yeah, very good question. Very good question again. So as I told you that I will answer few of your question. Like then one thing that if you ask me like next 3-4 question, two things might happen. One thing, it will be beyond like discussion, like it will be more complex, and the other option is also true that I am not also aware. Right? Both are true actually. I'm honestly saying that, but this question is very easy to answer, whatever you ask, and you will understand that. So, for example, you have this mass of cell. This is actually, one textbook, like this slide we have before also. So, for example, this cell, it's express something, and that is providing the extrinsic signal. And it depends. It will only convert cells which are present nearby, like 2, 3, 4 cell nearby. Although this cells is exactly similar to, like, although the cell present here, it is similar to, this cell and these cells and these cells are same, but this signal is not reaching here. So as a result of that, it will only affect cells present in the nearby, and this will go into a different direction, and this will go in a different direction. So that way, this is, that how far away the stimulus is present or the signal is present. Based on that also, it can divide, or it can, decide the fate of those cells, the concentration of that stimulus. So this is very complex. So as you understand, I'm just giving you one example. I believe you guys know in many animals even that whether the animal will be male or female after birth, that depends on temperature also. Are you aware of this? Most likely from general knowledge question you are aware, right, Rajesh?
Yes, sir. In
Yeah.
mostly
So
reptiles, I have heard.
mostly reptile, right? So that's why the system is very complex, and I completely accept, Tanmay. So whatever you ask, this is good question. I was able to kind of like answer this question. But if you ask me question next, then I will say I don't know, because I think somebody who is more expert in developmental biology, that person will answer you more. But finally, we have to say no, we are not aware about this, because this is really a complex thing. But that way, finally, it works, and we get different cell types.
Yes, sir, I got it. My
Yeah.
question was answered.
So now the next step is like, as I told you, the like in a multicellular organism, like multicellular means we are multicellular organism. We have many cells. It's very important. So what is the major or the very important things to become a multicellular organism? You need to divide your cell, and those cells should adhere together. Otherwise, like bacteria will divide and it will make two new bacteria. But in our case, cells are getting divided, but still we are together, right? But in multicellular organism, the cell division and the cell death both are equally important. Cell death is also equally important. I told you before that cell death is very important, because during this division, if something major mistake happen, then what will be the thing with that cell? You cannot carry that cell. That cell will give rise to again another cell. That will be problem. So the thing is, that is called programmed cell death or apoptosis. It is an orderly cellular self-destruction pathway when DNA damage is not immediately repaired, or it is beyond repair. The cells kill themselves by programmed cell death. In absence of apoptosis, the cell can accumulate, and that can lead to cancer. So we will discuss a little bit about cancer also. So I'm not going into the details mechanism of apoptosis. It is not required for you. But what I want to tell you, that programmed cell death is absolutely important for normal health, as well as it prevents many diseases. As I told you, that if the system is robust and properly functioning, most of the cells with bad element, with lot of mutation, that should be discarded from our system. Okay, but apart from that, I should mention that they have some, like they have their role in normal physiology. I'm just telling you. You can see my hand. I have 5 fingers here, like. But when I was like very small, like few weeks old, even before birth, my hand was not like that. It has no finger. It's like this. It's kind of small palm-like thing. And then how this finger appeared? So that is again apoptosis is required. That's why we have different body parts, different thing. So here in this part I'm trying to explain you. So when kids is growing inside mother's body, say for example this part I'm zooming in. So this is like hand, and this will be finger later. So it is growing, right? During growth, what happened? Some cells get died here. Some cells get died here, died here, died here. Died means programmed cell death. This is programmed, not randomly. So as a result of that, again, when it is growing, then what will happen? This portion, some cell died. So as a result of that, this portion is increasing. This is increasing. This is give rise to finger. It is again growing. So this is just one classical example of how even apoptosis help to to make like different body parts, different system, right? This is absolutely required. Like, so I just gave you one example. There are many examples like this. So here you should remember that programmed cell death is also very important, just like cell division. Otherwise, if programmed cell death is not there, then we will have a big mass of cell like this. we shouldn't have this kind of different body parts. So during early stage of development, program cell that is happening constantly so that we can develop different organ with different shapes and size and all those things. Those are very important. Okay, and just a minute, it's like still it is not 10. So I will discuss up to 10:15. Okay, so please stay here because some interesting discussions are still left. We will the rest of the portion we will discuss about cancer mostly, and so that will be useful information. So let's see. I believe that you don't have class before 10:30, right? So you don't have class, right?
No, sir. No, sir.
Okay. Okay. So I will
After
go.
10:30.
Yeah. So I will try to finish by 10:15. Okay. So and molecular biology of cancer. Quickly, I will go through it. Not very much of details I will discuss. As I told you, even no one actually knows all the reason behind the cancer and how it works. Some things known, something are unknown. It's very kind of puzzle kind of thing, I would say, right? So still, whatever we can discuss in this class, let's see, right? So this is one cancer cell in the middle, a big one. And those small cell, those are some of our immune cell. Those cells are trying to destroy this cancer cell. This is some image only, nothing important here. So now this you already seen before in the whiteboard. I was trying to explain you. So in this context, in the cell cycle context, like we put this in terms of cancer. So here I just forget to mention one thing here. You guys know that some cells are not dividing in our body. Can you give some example? So here everything is moving, like the cells are getting divided, but some cells they are not dividing at all. So any example?
So neurons.
Neurons.
Yeah, mostly like
Neurons.
you will get very recent literature that might say that it divides, but those are at research level, few specific conditions. But mostly,
Sir,
yeah.
sir, in our brain we also have the glial cells as well. So the glial cells are also non-dividing cells.
Yeah, that's what I'm saying. Some cells, some brain cells, they also divide. That's what I'm saying. This is, the understanding our brain or nervous system is, more recent. I would say people are still working and many things are unknown. But based on the textbook knowledge, like whatever still people believe that most of the neuronal cells they don't get divided. The only thing is like whatever on baby they have the like neuronal cell in during adult time almost the number of neuronal cells same. Only thing when you see something, when you read something, we make some kind of connection between neuron, and that makes you to remember things, understand things. But in general, the number of neuronal cells is not getting increased over the time. Other cells, I told you that 10 to the power 13 cells, the cells are getting increased when you grow up. But as far as my understanding from the textbook, I never worked in nervous system or any neurological problem or anything. But the thing is, in case of nerve cell, they don't get divide. Recent literature, recent research suggests yes, in some condition it divides definitely, and I also agree with that. But still in textbook you will find this stage that is called G0 phase. G0 means from G1 it escapes and it's present somewhere here. It will not be divided anymore. So for example, some of nerve cells they present here. They are not going into again in the S phase. So as a result of that, this is called G0 or gap 0. Okay. And then and other things I already discussed actually. And here the cell cycle control, I told you when you drive your car, scooter, cycle, you should see the road signal. So cell is moving from M to G1, G1 to S, S to G2, and G2 to M. So during this time, there are many traffic signal, right? Some of the major traffic signal are mentioned here. Those are not traffic signal. Those are called major checkpoints. So where cells are properly checked whether they are fine or not. If not, then that cell shouldn't divide again. Rather, that cell should be actually, should be destroyed by program cell death or by other mechanism also, right? So as a result of that, those things are very important. But what happens when cell cycle regulation goes wrong? Because we are assuming that this is happening all the time properly, like all these things, all these checkpoints are properly functioning, and if properly functioning, then no mistake will happen, right? But the thing is, what happens when cell cycle regulation goes wrong? Then we will have uncontrolled cell proliferation. What is the outcome? Uncontrolled proliferation means tumor development, right? Tumor and cancer, they are almost synonym, right? So sometimes we say tumor, sometimes cancer, but that is okay. I'm not distinguishing them. So cell division without checking for DNA damage, right? So DNA replication error are not corrected, or DNA damage due to external mutagens are not corrected. Both can happen. Like during replication, some error is happening, or we are exposed to some radiation, some chemicals that are like not during replication, but that mutate some of our nucleotides that causes some problem in our nucleic acid sequence. So as a result of that, those are also mutation. Here you will see some kind of calculation that error rate of DNA polymerase, that means DNA polymerase synthesized DNA, is 1 in 10 to the power 5 nucleotides. That means while DNA polymerase is replicating our DNA, the error rate is 1 per 10 to the power 5 nucleotides. And a human cell has 6 billion base pair, as I told you before, in diploid cell. So one round of replication of the human genome will make that many errors. But then everything will be error, right? But that is not going to happen. Why? Do you know that? I mentioned during replication class that during replication, DNA polymerase has proofreading activity. Okay, they have proofreading activity. So although they do some mistake, but we have very nice proofreading activity, repair mechanism, then our DNA can be repaired again. And after replication, a well-designed DNA repair machinery present. But if the DNA damage is beyond repair, the cells die and initiating apoptosis. That means the cell will be destroyed. So is it clear? Like overall, I'm saying. If not, ask me. It's okay, right? So I'm continuing then.
Yes, sir.
Yeah. So now what happens when DNA get damaged? So there are many things that can create problem in DNA, but the cell cycle pauses as checkpoint, so cell will not divide. As a result of that, this whatever the misinformation, wrong thing, that will not be populated again because the cell is not getting divided. That is good. DNA repair program, we have very nice control mechanism that can be, that can help to repair our DNA. And the other thing is programmed cell death, as I have already told. So now, what is mutation? Mutation is, change of nucleotides. I'm not going into that details. I already told you, you can get this point. So now, this slide is important. So there are two types of mutation we are talking here. One is called somatic mutation, one is germline mutation. That means when this sperm and ovum are getting generated, this figure, I started the class with this cycle, right? During spermatogenesis, that means during sperm formation or during ovum formation, if some mutation happen in the sperm and ovum, right? What kind of mutation? Somebody asked me, I think Tanmay asked me, what happened during crossing over if something major, some major mistake happened during crossing over, right? So that you can also see germline mutation, some problem. So as a result of that, after fertilization, the zygote will be defective. And if that defect is really challenging, then that zygote will not lead into full-grown baby. It will not make embryo, and that is good actually. You know, the next generation with lot of defects, developmental defects, that is more problem, right? So as a result of that, if some mutation, some major problem happen, then the zygote will not lead into the next stage. That will not form this embryo, and as a result of that, that pregnancy will be aborted. Okay, so in this context, it is good. But in other cases, what happened, even after the synthesis of zygote, after the formation of zygote, at morula stage or any other stage, somatic mutation can also happen. Somatic mutation means what is that? During mitosis cell division, during normal DNA replication, some error happen, and for some reason, like our machinery cannot repair that problem. So that will accumulate some cells in that portion, some localized portion. So uncontrolled proliferation, that will make some kind of tumor growth. Okay, those are the possibility. And sometimes those tumor can actually invade, and it can be, it can be like those tumor cells are getting separated, and they are carried over through our blood vessel to other portions and of our body, and that tumor can go here and there. Normally tumor is localized. Where a tumor happened, that should be localized, that should be present there. But when this tumor or cancer getting distributed in the other part of the body, what we call that? You know, tell me.
Malignant tumor.
Yeah, very good. Malignant, metastasis, all we say. Like for example, many lung cancer, sometimes they spread, like many blood, like, so that is why malignant cancer or metastasis also, like both are similar thing. Yeah, very good. So as a result of that, although most of the time, if the germline mutation is huge, then zygote will not develop into embryo, but during somatic mutation, it can be accumulated and it can give rise to tumor. So here we are showing some, some basic outline of tumor development. Say for example, this is some epithelial cell, that means some skin cells or some layer like surrounding some of the organ like kidney or liver or anything like that. So when cell proliferation happen, during that time, because of some replication error, some mutation happen. And that mutation, neither, it is getting rectified, or cell division, different checkpoint is not controlling the cell. So as a result of that, cell getting again proliferated. So as a result of that, you will have more and more wrong cells here. And finally, those cells are again proliferated, and those cell masses, now what happened, they can be, flowing through blood vessel. You can see here that cells, they are forming some cancer like tumor stage here, and those tumors are now invading, and they are distributing through the blood vessel, because those cells somehow they have wrong information and our body cannot tackle them. So as a result of that, if those cells are getting distributed through blood vessel and accumulated in other portion of our body, and somehow it will create the problem in other portion of our body, and that is called metastasis. And someone you told already that malignant cancer, that means it is going here and there, and it's create a lot of problem. This is, I would say, at the very end stage of cancer, right? That will be very difficult to handle, or the handling this situation is very difficult. So, but honestly speaking, we have very good system. No worry. Whatever happening, whatever we are getting challenged every day, our system can take care of it. Like either this cell cycle control mechanism or DNA repair mechanism, all those things, they are taking care of it. Even after that, we have very profound immune system. Immune system also helps to check cancer. So only in certain people, when they develop cancer, many things that failed simultaneously. Yeah, but that's the reason, that's I told you before, that we don't know why all those checkpoints, they failed together in certain individual, right? That is the big question actually. I would say for example, on particular individual, that person is perfectly healthy, very maintaining like a very good lifestyle, like not doing anything wrong, like maintaining everything perfectly. And say for example, only after 60 years of age something happened and develop cancer. So how you will explain that? That's why I told you it is not predicted that much also. There are some prediction that if someone very much kind of smoking cigarette all day and night, that might cause more mutation in lung cells, and that causes lung cancer, right? So in the some slide I mentioned there some mutagen like tobacco and other things, but those you can predict. But also you will find sometime that although someone is very healthy, no problem, no like any other drug or anything, any bad thing, but still develop cancer. So that's why I told it's sometime it's not explainable, right? But those are the processes normally what leads to cancer. Okay, so
So,
by this, yeah.
yes, yes. So, like we learned that some immune system plays a role in taking care of cancerous cells. So, sir, why don't we as part of genetic engineering? Why don't we inject such cells when the cancer moves to the
Yeah,
third?
yeah, Tanmay. Yeah, I understand your question. I must say this is good question. Although the question is formed not in a proper way, but it's very good question. Yes, Tanmay, this is absolutely important question. Whoever present here, try to listen that uh, actually cancer immunotherapy is a very big field now. Many people are working and also people are using some therapy. I'm just giving you one example. Like one cancer is called melanoma, that is some kind of skin cancer, right? So people are using some immune therapy against that cancer, and people are successful. But those therapy are very costly nowadays, like radiation therapy, chemotherapy, whatever used. Those are like after chemotherapy or radiotherapy, the problem is the side effect is lot actually, because they are not specifically killing those tumor cell or cancer cell or malignant cell. They like their bad effect is throughout our body. But most of the immune therapy are directed, like they are specifically target some of those cells. And the good thing is, yes, that kind of things are already there in place and people are being treated. But the problem is cost is still very high. It is not very much feasible for all of us. But the thing is, in particularly in Western world, it is very common. And most likely, I forget the year. I would say 19 like 2 or 3 years back, two scientists they got Nobel Prize in their work in immunology field, and mostly their work directed towards this point, that how you can modulate our immune system for to fight diseases, either it is cancer or infection, right? So it is possible, Tanmay.
Okay, sir.
Okay.
Excuse me, sir.
Yeah.
Sir, is it possible that something happens and our immune system is not able to tackle cancer? So the tumor keeps on growing and growing, and that whole organism grows into a filthy mass.
Rajesh, can you tell me your question, please, once more?
Sir, is it possible that our immune means all of the cancer treatment method fails and the cells keep on dividing and dividing and the organism grows into a filthy mass?
Okay. So you are saying that cancer, like cell, the dividing, and it will make a big mess, or something like that.
Yes, sir. For simple organisms like uh, simple multicellular organisms like sponges or
Yeah.
starf. Yeah.
Yeah. So see, Rajesh. So actually, you will see many human individual. You will see like some portion of body, like from face, like it's kind of big bulb-like thing in the shoulder, also like big thing. So those are also some kind of tumorous growth, but those are benign. Benign means they are not moving from that portion. Like only they are whatever they are growing, they are localized in that place. Okay, that is not that much harmful. But what happens sometime, like, people try to operate those, and during the operation something happen, and that causes more problem. But those benign thing is okay, actually it will grow and it will stay there. But the problem is, those are mostly like, tumor, those are formed on the, like, on the peripheral part of our body. But if the same thing happen inside our body, like one, I'm just telling you one type of cancer, like cancer associated with our, like, kidney, very close to kidney. If this kind of mass of cells growing, then that will create problem, and that will make, like, the function of the kidney and other portion will be, problematic. And finally, if it is unchecked, then it might be, might go into metastatic stage also. That will create problem. Okay, but as long as, like, cancer, they are benign. Benign means they are not in metastatic condition. They are localized. It is easy to treat, and it is not that harmful. Okay.
Yes, sir.
Yeah. Okay. So thank you very much to all of you, and you may leave now. And so next week we will